to many places, but New York University, I think, is her home base. She's a licensed physician and surgeon, and she also has a technical degree, which explains her interests in a lot of mathematical tools and techniques. And she actually does have some roots in North Carolina. She went to medical school at Duke University after graduating from Dartmouth, and she is a very illustrious researcher in the whole field of placentas. I don't know what the exact term, medical term is. Placentology?
Yes.
It is?
Yes.
No kidding.
No kidding.
Oh, wow. All right, fine. And she's, as I said, currently a professor at the medical school at New York University. She's also director of Early Path Clinical Research Consultation Services, and she's affiliated with many different hospitals in the New York City area. And she has a knack at communicating with technical people, and basically, which gave her this very prolific and illustrious career in publication and in all her academic
Let's wrap it up.
career. It's a pleasure to have you here.
Thank
and she'll be entertaining us about measuring the placenta. Thank you.
Two things. One, I have a knack for communicating with technical people. Now I have major performance anxiety. And two, I'm going to entertain you to very high bars that I do hope I will try to meet with a subject that is most people don't care too much about, including obstetricians. But the placenta is what was attached to your belly button before you were born. And the placenta, before you are born, serves the function of your lungs, serves the function of your gastrointestinal tract, serves the function of your kidneys, in that you get your oxygen and nutrients exchanged from your mother's bloodstream across the placenta, and you get your oxygen through the placenta. Food comes from the mother's bloodstream into the placenta and then goes to you. Wastes are excreted from your bloodstream across the placenta into your mother's bloodstream. Most hormones that are made by the pituitary in the brain and in most of our endocrine system are also produced. If they're not produced, releasing factors are made by the placenta. It's really a great little structure, and as soon as we're born, we're cut off it, and most of the time it's thrown in the garbage. But I'm going to try to tell you not only why this is important for a healthy life before birth, so that you can start off life reasonably intact, but it's becoming increasingly important to understand how and when in pregnancy things happen that can change not only the course of that pregnancy, but there's increasing evidence that what happens before birth actually colors the rest of our lives. So let's go through that. And I love the technology here, but I have to be able to, there, okay. Life before birth, or intrauterine life, is associated, if you have problems with this, you can imagine that you're at increased risk for dying, and you're right, if I touch the screen, it advances. Morbidity means sickness, and mortality means death. So dying before birth, or having a disease before birth or shortly at the time of birth. Neonatal is defined as the first month of life. Childhood, there are a number of risks of problem pregnancies, for risks for asthma, for cerebral palsy, for many neurodevelopmental, cognitive, and motor problems in childhood. Okay, and recently, over the last 15 years, i.e. less than the total time I've been practicing medicine, there's been a new data accumulating that suggests that life before birth actually is associated with health risks at age 40, 50, and 60. And that is a little bit hard to necessarily understand. How could that happen? I mean, once you're born, we're all, all created equal, right? So we all have a clean slate once we get out of the room, out of the womb, excuse me, out of the room of the womb. But basically, what this evidence sums up to is that as best as epidemiologists and biostatisticians can try to adjust mathematically, with confounding and all of the biostatistical methods, as to the best of our ability, once we try to correct for or adjust for everything that we do after we're born, plus our genes insofar as they're reflected in family history, a wide range of adult health risks, and this is up to from 50 to 70 is where most of the studies have been done, vary either directly or indirectly with birth weight. So either the higher the birth weight, the greater the risk, or the lower the birth weight, the greater the risk. And this is, okay. And genetics aside, 80% of birth weight is mediated via placental function. Bigger people tend to have bigger babies, and the placenta is 99.9% baby tissue, 99.9% baby. So it grows proportionally to, the genetic capacity of the fetus. And almost all of 80%, 4/5 of birth weight variance, apart from your parental size, depends on how well or poorly the placenta is able to establish itself. Now, what types of health risks are associated with birth weight? Are we talking about disorders that affect 1 in 150,000 people, very, very rare, obscure, and bizarre issues? Here I just hovered my hand over the screen. I'm not even touching the arrow. I have an aura. Cardiovascular disease. We're talking really about very common diseases. High blood pressure, stroke, myocardial infarct, which is a fancy term for heart attack, diabetes and related syndromes, including obesity and syndrome X, which is also called the metabolic syndrome, which is a major risk factor for obesity, where you carry all your weight in your tummy, and which carries high and independent risks for cardiovascular disease. So these two circumstances are actually more closely related than they might appear. But then there's also cancers, including prostate and breast cancer. And I am appointed in, of all things, a psychiatry department. People who know me well have suggested that I should be a patient of, not a faculty member in such a department. But CP is an abbreviation for cerebral palsy, ADHD, attention deficit disorder, and autism and schizophrenia. Now, these latter two disorders are strongly genetically influenced. The risks of having a second autistic child, or once there is a family history of autism, is as high as 10 to 20%, whereas the risk in the population as a whole is less than 1%. However, in both autism and schizophrenia, despite this genetic predilection, there seems to be something built into the families. The children who tend to manifest either of those two conditions tend to be the ones who were lighter, born preterm, have their cord wrapped around their neck a couple of times, etc. So people who had some type of complication during pregnancy. Now, cardiovascular disease and diabetes. With cardiovascular disease, your risks of dying at age 50 of a heart attack are less if you're 10 pounds than if you're 9 pounds, less if you're 9 pounds than if you're 8 pounds, etc. Works right across the entire normal birth weight spectrum. So it's not just like the little itty bitty babies in the intensive care unit are causing all of these problems. These are circumstances, and the idea of programming your biology before birth is something that would apply to everybody in this room, regardless of your mother's pregnancy history. With diabetes and related syndromes, again, certainly there's a genetic component to diabetes, but the idea is that your risk of diabetes goes up as birth weight goes down. And why would that be? Why would a skinny baby have a greater risk of developing diabetes? The idea is that if before birth you are attached to a placenta that is not working right, so that you are chronically hungry for nutrients, your physiology starts setting up receptors and thresholds and sensitivities so that you really are able to hang on and maximally utilize every single calorie. And you don't spend a calorie unless you absolutely require it. It has to happen. As soon as you're cut off the placenta and everybody wants to fatten up that skinny little kid because otherwise they don't look like a good mother, and they eat and eat and eat, they end up putting on weight, and then their biology is still set to optimally manage every calorie. So the idea that diets don't work for me can actually be physiologically it could actually be true for people. And one of the things that's of some interest is that there are risks that are associated with certain ethnicities. For example, African Americans have very high rates of hypertension, diabetes, obesity, etc. African Americans also have a very high rate of infant mortality and morbidity, about 50% higher than other ethnic groups in the country, matched for city, matched for neighborhood as best we can, what have you. And most of that increased poor newborn health is related to babies being born too small. So you can see that there's the potential with this type of circumstance that if a mother has a baby that's small, that child will grow up to be a sicker adult, may have more pregnancy complications themselves, and it's what's called the transgenerational passage of diseases. A problem happening in the grandmother may not be fully manifest as early onset disease until a granddaughter, as the effects of passing the predilection, passing the problem through a couple of generations becomes fully manifest. Now, for example, with cancers, breast cancer in particular, the bigger you are, all other things being equal, including your genetics, the greater the risk of breast cancer, and that's thought to be related to having an optimal placenta and an optimal endocrine and growth factor environment, so that there's some programming to the embryological mammary tissue, so that it's more geared up to be responsive to something that wants it to grow. So it has a greater chance of its growth becoming uncontrollable if something else happens to it later. So that's a little bit more than I intended to spend. But I think this stuff is really neat. And the animal models of all of these diseases suggest that it's not just size that matters. And of course, my standard joke, and you've heard it before, is that, of course, I would not want to think that being small was something that would be intrinsically bad. Okay? So the animal models suggest that it is not size. That it's not that if you're born at 5 pounds, you have a smaller heart with fewer cells in it, so that at age 50 or 60, you have a greater chance of it wearing out. It is not just size. It is something about either the network of vasculature that you develop, something about this physiology, etc. So it is not just size. So that makes me happy.
There's also the fact that nutrition, the mother's nutrition has a lot to do with also the baby's physical, at least appearance in terms of size and...
That's true. Of the things that influence, there's maternal age, socioeconomic status, parity, and what that means is how many babies you've had. Second babies tend to be bigger than first babies. Gender, boys tend to be bigger than girls. And then maternal pre-pregnancy weight, which is related to height, and then pregnancy weight gain. And again, when you put on the weight in pregnancy also is important, whether it's first, second, or third trimester. The thing is that birth weight, though, is actually quite you can't modify it very easily with vitamin or nutrient supplements. They tried to a study in Harlem in the 60s of providing people with more protein and what have you, and they weren't able to budge birth weight more than half an ounce, by improving diet of, inner city, low-income folks. And so yes, these large things do influence birth weight, but things like just nutritional status per se is less important than you might expect. Yes, sir?
So even given ideal gestation conditions, minority people can't improve birth weight?
Even given if you the studies suggest that even if you have two medical school, a black classmate of mine at Duke, who had the same socioeconomic status, had the same neighborhood, who had the same diet, who had the same everything, and the same family history of other things, that person would have a 50% greater risk of infant mortality than I. Okay? So it's not anything that you can point to any one person. This is a population characteristic. But overall, that population, adjusted for all other things, has a very stubborn increase in infant mortality. That initially, for example, I went back and got a master's in biostatistics at Columbia, and one of the things that I did in the summer was I did an internship at the public health department in New York City. And 15 years ago, they thought that the increased morbidity and mortality in African Americans was due to poor access to health care. Okay, so they put in this enormous drive to make sure that everybody who wanted to go to a doctor could get to a doctor as soon as was humanly possible. 15 years later, they look at their numbers, and the rates of early onset prenatal care, before 12 weeks, how many people see a doctor, same across the ethnicities. All of the infant mortality rates are lower than they were 15 years ago, but still African Americans are 50% higher than the other ethnic groups. So I sat through a meeting where it was suggested that the quality of the health care must be different, and the providers in New York City must be intrinsically racist. I don't know. But, these are alternative explanations, and I think that understanding this better takes it out of philosophical arguments, and let's figure out what the heck the biology is. Is there a biology? Then let's just deal with that. Now, how do we measure placentas? And this is an artist's rendition of an umbilical cord. And this is the placental disc. And placenta, I forget if it's Latin or Greek. It must be Greek. Means little cake. Okay, pathologists are supposed to be really renowned for disgusting food analogies. I try to keep mine to a minimum. But how do we measure them? Basically, the same way we've done since 1961. In 1961, a former Luftwaffe ace, who is the great-grandfather of placental pathology in this country, Kurt Benuschke, wrote a protocol, and that's exactly what we're doing now. And in fact, the government is in the process of committing $4 billion to a project called the National Children's Study. If you put that in Google, you can find out about this, where they're going to follow 120,000 pregnant women to delivery and follow the expected 100,000 live-born babies to age 21 to better understand these fetal origins of health risks. And many of my colleagues are thinking that we should do it exactly the same way we did it in 1961. But luckily, I'm working with some people, and we're going to try to implement some of the new measures that I'm going to talk about with you today. But we're going to change that, right? So why do we need better measures? And let's see if I can make this there. Oops. This is probably not going to animate too well on this. But if you measure something poorly, right? Say you measure the placenta poorly. And what you want to understand is how maternal smoking affects, this is sigma birth weight, variance in birth weight, just as an example. Could be childhood IQ, could be anything at all. If you measure the placenta poorly, then you're going to have an attenuated relationship between the maternal factor and the placenta, because this is going to be fuzzy and amorphous. And then what's going to happen is that it will appear statistically that the maternal factor affects birth weight directly, and bypasses the placenta totally. However, if you measure placenta better, then what happens, you might be able to find that the true relationship in here was not this, but that the mother affects the placenta, and by affecting the placenta, affects the baby. So if this is not the case, but this is, say it's smoking, say it's an environmental exposure to arsenic. And I worked in Chattanooga, where 80% of the placentas in a 3-month period in Chattanooga, Tennessee, had detectable levels of arsenic because of groundwater contamination and pollution in general from factories. This allows you to approach possibly intervening and dealing with things in a completely different fashion. Maternal stress, any type of factor. So what we're trying to do is understand the biology. It is really important not to measure our intermediate step poorly. We need to measure the placenta as well as we measure birth weight, as well as we measure maternal smoking, what have you. It has to be well measured, or else we're going to get a false finding. Why is this whole problem important? Well, if we can measure placentas well and take measurements of the placenta at delivery, what we can do is understand when in pregnancy a developmental trajectory started to go awry. For example, I'm looking at all of you now, and I've never met most of you before right now. And I certainly wasn't looking at you at 5 or 6 weeks when you were in the womb, but we know that your neural tubes, you know that you initially, your brain is like a flat piece of paper and it turns into a tube and zips down the middle and seals at both ends and becomes your brain on one end and your spine on the other. Pretty amazing stuff. This is why I do what I do, because I just love embryology. But I know that that happened to all of you. And early on we have a little flipper. And at a particular, within about 12 hours, all of us at the same time in our gestation, cells started to die along 1, 2, 3, 4 lines in that flipper. And that's why we have 5 fingers. So I know that you had all of these developmental steps happen, even though I wasn't watching you at the time that it was happening. And the placenta also is the accumulation of a series of growth and developmental trajectories. And if we can measure it carefully enough, that's when we're going to get into when. Why is when important? Because the earlier the stress, the greater the risk of fetal effect. For example, if you do something that kills 1 cell at 8 weeks, that if it had lived would have given 500 cells by term, killing 1 cell at 8 weeks is a lot worse than killing 1 cell at 40 weeks because you lose those 500 cells. Okay? So the earlier the stress, the greater the risk of fetal effect. If we could understand how severe that deviation from growth trajectory would be, again, the more severe the stress, the greater the risk of fetal effect. A gentle stress is something that you can habituate to. I used to say that this was like being married, that after 15 years you'd say, you know, I never thought I would tolerate this kind of not, but, you know, it's a slippery slope, you know, I mean, all of a sudden, here I am, you know. But if something happened that was very abrupt, hey, that would, you know, then you would notice it, and physiologically you would have a change in homeostasis, possibly. So if there's a severe effect, you might have altered homeostasis compared to a milder effect, which you might be able to accommodate for without really any type of biochemical changes. And again, the greater the fetal effect, the greater the potential for long-term modulation. It may be that some of the measurements that we come up with may say that the developmental trajectory was not just modified at one point in time, but that it was modified at several points in time. If this is a normal developmental trajectory, that maybe somebody would, and that would be one hit, and then the developmental trajectory would come up. But maybe for some people, you know, that there are multiple hits that bring them down from baseline. And this may be very different physiologically than someone who's either programmed to be small, okay, or chronically stressed from the beginning, small from the beginning. So these types of information, when, severity, and single hit versus multiple hits, all could give us information that would help us understand the fetal environment better and be able to get some biology. So again, if what we're trying to do now is understand the nature of shape variability, and also branching, why I'm here is because your group and working with Jamila has given us the possibility that we may be able to develop metrics that would help us quantitate and apply standards to branching variability, and again, with the idea of understanding better intrauterine life. Now, what are the types of measures? I was told that saying that there's a gross examination is really not a very, the colloquial use of the word gross, is disgusting, right? It's gross. And placentas are really bloody. And that's when I started dressing out of secondhand stores, when practically whatever, but it's a naked eye examination, okay, looking at the whole placenta. And the whole placenta is what we're going to be focusing on here. Then there's also microscopic pathology, and microscopic pathology, here's a naked eye placenta, and you have an umbilical cord insertion here, and you can see that this is a little bit of an irregular, you're going to be seeing this placenta a lot, but this is a naked eye photograph of a placenta. And then we can, I don't know why that erased, and I don't know, I'm being impatient, aren't I?
Well, that thing does take
Okay, there were some slices in
you
there.
Can actually, you can use your mouse.
Okay. When you take this, you slice it. And you look at the thickness, okay? And the thickness is actually where the maternal blood, this is the part of the placenta that's attached to the uterine wall. So the mother's blood vessels are coming up and feeding into the placental tissue here, and this is actually where nutrient exchange is taking place in this dimension. Then what we do is we take a small square out of it and make a glass slide that we can take and put under the microscope and review as well. So thanks for the tip. Okay, what I'd like to talk about are the measure type, the current way it's done, the limitations of how it's done currently, and a little bit about our work in moving from structure to function, and I'm probably going to speak progressively faster as the minutes pass. This is whole measures, okay, naked eye measures. And the current way that we measure the placenta is a single length, width, and depth. So we take the larger diameter and we take a smaller diameter, and there is actually no standard that requires those be at a perpendicular. I tend to. And then you measure the depth of the placenta, and the more normal a placenta is, the more uniform the perimeter, the more uniform the thickness. And basically these standard measures work pretty darn good for placentas that are from healthy, normal uterine environments. However, this placenta, I told you you'd see it again, this placenta is obviously very irregular. And this placenta clearly has issues with how it was growing, in the womb. There's a lot of variability there. What do you do? Do you take the maximum? Do you take the minimum? Do you average the two? Do you take a majority percent? How would you get one number that would characterize this placenta? Now, the placenta normally starts off with an umbilical cord pretty much smack dab in the center. All of our belly buttons are pretty much in the same place, right? And this is I love embryology, but when we're a flat piece of paper, our butt is down here, our spine is here, our brain, then our mouth, and then our heart. And our belly buttons are the result of us folding top to bottom so that it's heart, mouth, and brain. And then side to side, so the belly buttons are where those two planes of cleavage, of folding, intersect. And so early on, the placenta also has that type of structure, and its belly button is where most of the major big blood vessels are coming out. And if you are going to be, this is going to come into something that we're going to talk about at the end, if you are going to be devising an organ to transfer oxygen and nutrients, what would be the optimal design? Well, it would be probably radial, right? And it would be equal length of trips, right? And you'd have, if these are the nutrient supply areas like the farmlands, every nutrient supply area would not be too terribly far from a blood vessel, etc. So there's an optimum transport component to this that would dictate that these placentas be reasonably uniform in thickness and reasonably uniform as far as growth about the umbilical cord and its perimeter. However, these placentas obviously were changed. You know, these are not round to oval. The umbilical cord is not in the center. The umbilical cord is actually over here. And if early on the placenta was centered about the umbilical cord, well, early on, maybe half of this placenta got killed. Women often, not uncommonly, have first trimester bleeding. Maybe there was an event that killed a portion of the placenta to result in the umbilical cord being marooned at one extreme edge of the tissue. And again, just like this implies something about the intrauterine environment, the placenta will arborize. This is all branching growth here. What makes the placenta thick is a series of dichotomous branches, okay? And it will branch where the blood flow is lush and gentle, and it will not branch where the blood flow is maybe too harsh and damages the tiny developing tissues or what have you. Where the uterine lining is not good, it won't develop. So basically, at some level, you could consider these two dimensions, if you could understand them, you could, and I always still sound like I'm faking it when I use math terms, because I have one of the worst cases of math anxiety you'll ever meet. But the inverse solution to this shape would be the intrauterine environment. Okay, because the intrauterine environment modulates this dimension and also modulates this. If you could solve for the two of them, you would be able to describe the intrauterine environment that created this and deformed it. You might have multiple solutions to this and multiple solutions to this, but my hope would be is that if we put the two together, that you might end up with a much smaller family of solutions that would account for both of them. And then those solutions would differ possibly by the time of onset of the stress, the severity of the stress, the multiplicity of the stress. And the reason that I walk around and still go to hospitals and see placentas is that there are a lot of obstetricians who want me to help them understand when they deal with somebody who has a complication at 14 or 16 or 18 or 20 weeks, they want to know how that baby turned out. So what we can do is, if I have a series of hypotheses that are the unique group of solutions that both solve this and this at the same time, that generates a set of hypotheses that then I can start looking at people throughout pregnancy and start seeing which one actually is, do they all happen in some people? Or is there one solution that really is the most common one that we see in humans? One pattern of severity, multiplicity, and timing. Or, is one of those solutions work for one set of people? Maybe it would work for people who had had low socioeconomic status, that there would be one pattern of pathology that would be more common there. Maybe there would be no distinguishing features. Maybe only one solution would really be. But that's the kind of work that we're looking to do. Now, the other problem with measures is something, again, the main reason that we're here and talking to Jamila, this is a branching organ. And here's the umbilical cord insertion on these two placentas. And I think you can appreciate that here there's blood vessels and blood vessels and blood vessels. There are lots and lots of blood vessels, and they're doing a lot of branching. This is a very complicated chorionic plate surface. And I can say that it's more complicated than this one, which only has really three main branches, okay, and goes for long stretches on the placental surface without branching. This is very different. We don't have a metric for saying how much different this is than this. But certainly from the point of view, if this was a railroad, if this was a railroad system, as far as efficiency and cost, these would be very, very different functioning entities, okay? Why do you generate this? What are the circumstances that cause this to be generated? Then what is the impact of having a more costly system on the baby? Because remember, how does this whole system work? I love to draw. You have a baby, and the baby has a heartbeat, and about half of every heartbeat goes down the cord to the placenta. 50% of every heartbeat, okay? So the heart pumps the blood down, and there is no pump, there's no heart in the placenta. So basically, it's the force of the heart that gets it all the way down into the capillary bed where blood moves very, very slowly, right? It moves faster in big blood vessels, very slowly in capillaries. How the heck does it get a head of steam to go all the way back up the umbilical cord? It's still the pressure of the baby's heart with the pulsations in the umbilical cord. What the baby gets out of every heartbeat cycle basically is the amount of nutrients transferred minus the cardiovascular work. Okay? So if you have an inefficient system, this number will go up, and presumably the fetal benefit from every cardiac cycle would go down, and this would modify birth weight. Remember, birth weight is what is being used in all of these studies as a proxy for intrauterine health, because these studies that were done on 50, 60, 70-year-olds, they did them on people who were born in the teens and 20s. And they found places, for example, in England, Herefordshire. Where's Herefordshire? I don't know. It's a little place where midwives wrote down the birth weights. And there were enough people still living there 50 years later where they could go back and say, you were this much weight when you were born, and they could match it up and look at the disease. So this is just a proxy for intrauterine health. Yes.
Do we know the weights of the babies born to these placentas, or are these just detached from the baby's kind of observational placenta?
Okay,
this is part ahead.
No, this is part of a cohort that was collected down the road, University of North Carolina. They had a contract to recruit people at age 18 weeks before it was known how they were going to end up. And they measured them and asked them questions and took samples and followed the pregnancy. And then whenever they delivered, the placentas were shipped to me, actually to my home. My family used to love the boxes of placentas. They rarely leaked, but in any case. And, this is a study ID number. I know the birth weights on all of these, and I'm going to show you some of the, Simon and I have been working with this data set, and, Jamila will be working with that as well. Our hope might be that if we can find things that are predictive and useful, that this population could then be written up for federal funding to follow these children into early childhood. There's 1,200 people in this cohort. That's not probably enough people to study autism, but it would be enough to study blood pressure, childhood growth trajectories. It would be enough to study a number of different things, not rare disorders, but a number of things. Okay, and here again is our nice normal placenta with all the branches, and here's the umbilical cord. And basically, if I were going to trace around where the blood vessels are on this placenta, that's where they are. There's only one artery and one vein, and that's it. And the placenta, what is this placenta out here doing? How is it structured? What would be the cost of having a lot of major highways only in one spot, relative? What is the cost? So again, I can say that these two are not the same, but how much not the same? Is this one worse than that one? I don't know. So metrics, we need metrics. So, and again, chorionic vasculature, how long the blood vessels are reflects the number of mitoses or cell divisions, right? And if you're healthy, your cells can divide well. And if one of the things that can make you not healthy, my BlackBerry hasn't rung all day, but it's ringing it's ringing now. It is something about the welfare and the well-being of the intrauterine environment. And then vascular branching, specific genes control branching of vasculature, but the same gene families that control branching in the placenta also control branching in fetal organs such as the lung, the pancreas, the kidney, and of course, neurons with their dendritic arborization, which is essential for neuronal connectivity, and all of our complex neurological function is due to the branching density of dendrites. So the other thing is measures are not taken in relationship to each other. We have a larger and a smaller diameter, and then people measure where the umbilical cord is to the closest margin. And what pathologists generally describe things, the reason that you have a description on a pathology report is because the doctor wants to try to communicate what they receive. And basically, you can't form a mental picture of what these placentas look like from these descriptions. So they're taken completely as independent measures and not taken as being measures of the same structure that might reflect a shared biological health or pathology. So for better measures, what we did is we took photographs of these placentas, blew them up, put them on cartographics tablets, and marked them at least 1 centimeter intervals around the cartographics tablet. And perimeter and related geometrics, there's a number of different ways that you can look at ruffling and irregularity of the perimeter, accounted for just 36% of birth weight variance. That's a little bit more than a third, but actually if you just threw the whole placenta on the scale, it accounts for about a third of birth weight variance, just the weight. You see, we have a lot of birth weight variance that we need to explain. So basically, just this perimeter was as useful in explaining birth weight as the entire mass of the placenta. So we actually think that we're probably going to get we're going to probably get close to that 80% when we put all of these things together. Now, chorionic vasculature, this is Simon's a hand mask tool using Adobe Photoshop. We still have to, we've done a lot better with taking these structures out. Initially, what we had thought about doing was, overlaying a spiral grid and doing a stereological estimate as to how many times the blood vessels crossed the grid, and that would give us an estimate. But now with better image processing and the help of, friends, we're able to extract this. We still ain't got no metrics to describe what is normal. Here, obviously, there's more branching. Here, there's very little branching. This structure here, there looks like there's holes in this, where they just didn't cover the structure well. So we don't have any metrics for those. But we're better at extracting the information, so now we're ready for metrics to be generated. And then in terms of things being related to each other, the next thing we did was we took the perimeters. And remember I said earlier that you can sort of see where these blood vessels disappear. And what we did was we just marked where every blood vessel went away. Okay? And then we ended up with an area, an achieved area, a vascular area, and the umbilical cord insertion. Okay? So now let's take all of these things and try to look at them in relationship to each other. When we looked at this, when I did this, my hypothesis was that vascular area over area was going to influence birth weight. That if these blood vessels didn't go out very far and this placenta, didn't have a high capacitance, low resistance distribution system, a big blood vessel nearby, then this was going to be inefficient, and that these babies were going to do worse if this area ratio was off. So that was my hypothesis. the way we approached it is from this drawing, we can actually get 3 areas. One, the perimeter, which I've talked about already. The second, the vascular area. And then the third area that we can get is actually the area of a circle with the ideal placenta. The placenta that would be circular that had the achieved placental area. Okay? And each of those would have a centroid, a weighted center of the shape. And we looked at ratios, and then we looked at these centroids. And actually what we found out was if this is, that ideal, and this is the actual area, this is the ideal circle, this is the actual area, and this is the vascular area. What we found is that the ratio of these 2 areas was not predictive of birth weight, but how misaligned these guys were actually influenced birth weight a total of about a third of a pound. And as I said, that doesn't sound like an awful lot, but when you think that taking prenatal vitamins to not taking prenatal vitamins to giving people extra protein when they have adequate calories, that does barely a half an ounce to an ounce. These are actually relatively large amounts. So this type of misalignment implies that there is something about the growth of these structures. The greater that they are off kilter, what was the word that you used a word that I was going to try? That they are not concentric, that they don't overlie each other, that the umbilical cord is off. If this is the true umbilical cord that would be in the ideal circle, the distance between the true umbilical cord insertion site and the weighted center of the area, the distance between the umbilical cord insertion site and the centroid of the vascular area, that all of these being misaligned makes a placenta that this is all adjusted for placental weight. So for the same placental weight, the greater the misalignment, the smaller the baby. Okay? So we're interested in capturing these surface branches. We're also interested in capturing the branching of the three-dimensional placenta as well, but what have you. So placental thickness, as I've mentioned, is where actually nutrient exchange occurs, and this is a high magnification of part of this thickness. The mom red blood cells would be here. These are the placental villi. These are the cells that turn the pregnancy test positive. These make HCG, which is what's measured in the pregnancy test. And these are the baby's blood vessels. And you can see that there's a very narrow distance between the baby and the mother, so that diffusion works very, very efficiently to transfer. Okay? So that's how all of this works. So the work that we still need to do is figure out what are the metrics, and then what are the parameters that can be quantified by those metrics for our chorionic vessels now that we're able to extract them. And all of the work that we've done so far has been stuck on the 2D, but if we can take this thickness and consider it as a height function and add it in, again, I think, whether we solve these 2 equations differently and then look for the equations that solve, we look for the intersection of the 2, or if we simultaneously solve for them and integrate it with a height function type of thing. That's what we're looking for to explain the complexity of the shape of the placenta. And the shape of the placenta is basically the shape of the branching structure that developed throughout term. Now, we're interested in anatomy because anatomy has a function, and in the placenta's case, there is only one reason why you have a placenta, which is to make as much baby as you can. So what we're looking to do is to derive anatomical measures that imply functionality. So here we're going to talk a little bit work that we're trying to move forward on to move from shape to a cost function. And Ja, I don't know his last name.
That is his last name.
Okay. Apologies. I have good friends in Beijing, and I never get name order correct, and I apologize. He's at UCSD, yes, and he's been working on leaves. And if you give him the shape of a leaf and where the stem inserts, he can tell you what is the optimal transport function, what is the optimal distribution of, veins in the leaf to cover that shape. And from a placental point of view, surprise, surprise, the optimum transport distribution network is a circle that looks a little bit more like this with a centrally inserted umbilical cord. And the efficiency of this structure is around 6-7. So far, the only thing that he's worked with is the part of that graph that I showed you, the plots of the perimeter. So we've given him the shape of the placenta and the umbilical cord insertion. And what he does is he calculates the optimum distribution system for that shape given the umbilical cord insertion. And what he can do is say how much this shape and cord insertion what is the cost of having this shape and cord insertion given an ideal, an optimal vascular transport system. Okay. He hasn't looked at the vessels because we can't give him the vessels to look at yet because we have no metrics. So what he's looked at here now is this is the efficiency of the transport system, and this is a projected birth weight based on some calculations that he has done. And basically, the babies that are larger than are estimated have optimal efficiency. Okay, that this isn't the way I would show this data. I would take the observed birth weight, and divide it by the estimated birth weight and show that difference. But basically, he's finding a very high correlation between his measures of efficiency just based on these two data points, the perimeter and the cord. He's finding a high correlation of that efficiency being correlated with a bigger baby than you would otherwise predict given the size of the placenta. Okay. And, obviously we can do a lot more. Now, an hour's up, right? It's 1:30. Yes,
I had
sir.
a question about that efficiency plot and how it exceeded 1.
Okay, you're talking about these outliers?
I would assume that the maximum efficiency
Oh,
you could rate at a placenta.
that's a
would
cost. That's a cost function.
Oh, it's a cost function. Okay.
So maximum efficiency is that circle, and the way he calculates it, it comes out to be about 0.67.
Okay, so that's ideal, and then everything else is non.
Right,
Okay.
and what these placentas are, for example, is instead of a nice round placenta, a placenta that has a lobe here and a lobe here, and the umbilical cord is inserted here. Okay? That's extremely inefficient and very costly. Okay? So that's actually primate placentas are bilobed. Baboons, chimps, monkeys are multilobate, but human placentas are single lobes. Okay. What is our... I have 5 minutes. Questions? Should I skip to questions, or should I... Let's just... I'm not going to talk about this. We're doing some segmentation issues that are trying to help us distinguish better. Our bottom line is that microscopic review of these slides is very complicated, and what we're looking to do with image segmentation is improve reliability, have more aspects of a process like bacterial infection than just counting pus cells, which is what's done now, and instead of having things be semi-quantitative, some, none, some, more and a whole hell of a lot, which is basically what a 0 to 3 scoring system is, which is what was recommended by the Society for Pediatric Pathology, to generate continuous quantitative scales, because the disease processes that we're dealing with before birth that are associated with poor outcome are actually physiological continuous variables. And under the microscope, nobody has even tried to categorize placental architecture. And I'm going to skip this. It is pretty neat. I think that, you know, I obviously love what I do. And let's structure and function. The last thing, this was from IPAM as well. I was dealt with my math anxiety at the Institute of Pure and Applied Mathematics at UCLA for 3 months. Between April and June, and Denis Grubenkov and Bernard Sapoval at the Polytechnique in France are looking at how lungs function. And for them, this is the lung at rest. And at rest, we're breathing relatively gently. We're not having a great deal of force bringing things in and out. And there is not a uniform oxygen level at all parts of the functional unit of the lung. However, at exercise, and you start, you know, your chest starts to heave and what have you, there is a uniform, so this is exercise. And to be able to go from this to this is your pulmonary functional reserve. That's what allows you to exercise. And they looked at this from the point of view of conductance to reach the surface with the conductance to cross it. And if the reach versus cross, if reaching is greater than crossing, the surface works uniformly. If it's the reverse, then there's diffusion screening. And they calculated something of diffusion versus conduction and called it lambda. And they found that if they took a histology section through the lung and just traced the outlines, that the length of this line was proportional to this lambda. Now, we can do this very easily with the slides that we have, tracing the perimeters of the villi in histology. And what we are thinking is that in the placenta, the normal circumstance is exercise. The mother puts 20% of every heartbeat into the space around the placental villi. The baby puts half of every heartbeat down into the placenta. This is an exercise situation. But in diseases, maybe you have overgrowth so that you can hardly see where there would be space for the mother's blood to flow around this structure because there's so much of it. This is a diabetic placenta. So is there baffling and a limitation? Is it harder maybe for some of these blood vessels to see nutrients because there's too much placenta and there is screening and a less efficient placenta for unit size? By contrast, this is a placenta from a lady who became severely hypertensive, and the only cure for her was to take the baby and the placenta out. And Hippocrates recognized this disorder, called it preeclampsia, because if you didn't get the baby out, then she often seized and died, and the placenta out. And the treatment for this condition in the time of Hippocrates was delivery of the baby in the placenta. And guess what the treatment is now? Delivery of the baby in the placenta. Okay? But this is what the placenta can look like here. And here again, you have where are all the villi? You know, where have all the flowers gone? Where have all the villi? Why are there not more villi here? These are really big spaces. There are gaps here. And the villi that are here are really, really teeny weeny. Okay? So again, a disease in which you have abnormal function and diffusion screening, even within a placental functional unit, these are what the villi look like. And you could trace their perimeters, you know, like this. But then within the villi, the blood vessels tend to be relatively large and occupy the majority of the cross-sectional area. In disease states like preeclampsia, you have tiny blood vessels and not very many of them, And instead of them being located right out here so that there's good diffusion, it's almost like they've been beaten up, and now they're protecting their blood vessels by keeping them deeper down, because if they're at the surface, they would have been damaged and popped. And here is, again, a diabetic placenta that has way too many blood vessels, and some of them are out at the perimeter. But why should the baby want to have to pump blood through all of these blood vessels that are so far away from the maternal bloodstream that there's unlikely going to be any type of nutrient and gaseous exchange under normal circumstances? Okay, so these are the types of things that we're looking at. We like their metric because this is an extremely simple metric that we think we might be able to reliably capture, and that might give us some sense as to structure and function. And then the group that, once we have these metrics, what am I going to do? Am I going to try to get a birth cohort and then wait for 21 years until they grow up and what have you? I will be 72. I don't know, I think I want to retire by then. But in any case, they have a group in England that collected 12,000 placentas, and for 17 years they've been sitting in formaldehyde in Bristol, UK. And their executive committee gave permission for us to apply the metrics and apply our techniques to their studies, to their placentas, where they have followed these kids. They followed their growth, they followed their blood sugar, they followed their white blood cell counts, they followed their body mass index, they followed their cognitive and motor development, and this is where we're going to be applying these metrics too, so that hopefully within my professional career we'll get a little bit more understanding of this. Now, I know, so our vital organs depend on branching. The placenta is the only one of those vital organs that we can actually take apart. And our working hypothesis is that deviations from normal patterns of branching will explain health risks associated with complicated pregnancies. And what we really need is a way to, we need a metric system. This is a completely open area. This has not been approached at all in any way, shape, or form. And again, our goal is to not only understand how placentas grow and develop, but to potentially be able to use that to, at the time of birth, triage children for risks that might allow them to have access to earlier interventions and optimize their lifelong outcomes. Okay, thank you.
Okay, I want to give the chance to the remote site participants to ask any questions, if any. I guess today, normally we should be able to see them over there, but today, David, okay.
I cleared out the room. Never mind.
No, actually, because he went over. That's all.
All right.
Anybody? All right. Okay. If there are no questions, the remote site, let's get back to questions here.
Yes.
I would like to ask you if in the future would you envision a rating system just like they do on newborn babies, the APGAR score given to the baby? Would they give us a kind of a capture score for the placenta as well?
Yes, and that's one of the reasons why we've tried to keep the data collection methods very, very simple. A digital photograph is something, if you can take a picture of your kids at the beach, you can photograph a placenta. You know, we use the same camera. And not exactly the same camera, but honest. And the slides that we are using to develop the branching and segmentation, they're not fancy slides. They are routine stain slides that any hospital in the country and most hospitals in the developing world can make for a few dollars. So what we're trying to do is make sure that you don't have to do a $500 immuno special stain with all of this fancy stuff. That is, we'd like this to be useful, and that would be the use that we would hope that could be fashioned from it, yes. Did you have a...
Yeah, I was thinking, this may be a little bit detailed, but in one of your slides, you're comparing the centroid of 3 different kind of areas, and you're saying when they are most misaligned, then will most affect the size of the baby. Is there a reasoning or explanation for that?
Okay, I think, okay, this is my hypothesis, and I would like to be able to prove it, is that first of all, a deviation from this. And then you have the area, and then you have the vascular area, which is going to be smaller. And because we just marked down where the blood vessels go away, where it's jagged. So this would be optimal, right? So if the umbilical cord is here and the weighted centroid is here, what this implies is that the placenta initially was round, but its growth progressively became deviated, and the placenta that results is not organized as efficiently as it would be if it were like this.
It's not an isotropic.
Okay? And again, if the umbilical cord is here, or is here, for example, and the vasculature grew and developed, what's interesting is that this difference is important. This difference is important. And when you put them together, these differences each have their own predictive value. So there's something about the achieved area being off from the umbilical cord and this type of forcing the development. Early on, the major branches, there's 40 to 60 placental functional units. Those are thought to be established by around 14 weeks gestation. So if you've got 40 to 60, and early on they're radial, here, half of them may not have grown well at all or may have died back, and what you may have is hypertrophy, an abnormal growth of, so instead of them all being the size of a quarter, you have some that are teeny weeny, and then you have some that are really big out here, and that is an inefficiency as well. Okay? This I understand less well. Why these two should be completely independent, because you might think that this is going to be closely related to, the vascular area is going to be closely related to this area, but it's independent. And I don't know what it is about the stimulation of chorionic vascular growth so that this distance has independent predictive value. I don't know whether it's actually this area. I don't know whether it's vascular number. I don't know if it's branching density. I don't know. I don't have enough other ways of assessing the vasculature to know whether it's area or something else. So this one's a lot cloudier for me.
That looks like measures of symmetry. So if you can come up with a measure of symmetry, you'll be brilliant. Any other questions?
I also want to acknowledge the cultural impact of a research like that. When you announced that the effect of nutrition is half an ounce, but the knowledge about the structure before has so much more impact. This releases women from a big burden of being responsible from everything that goes wrong there, which is really a heavy burden.
Yeah, I mean, I do postmortem examinations. I talk to a lot of parents. And I was just talking to a woman last week who feels that it's all her fault, that her uterine environment was not a good home for the placenta, and her child died of chronic congestive heart failure because the placenta was so badly grown. Obviously, they want to know what happened. Will it happen again? How can I be protected? But I think that the ultrasound and modern medical technology has created the illusion that we actually see and understand. The approach of obstetricians towards my favorite organ is that if the baby is okay, the placenta's got to be pretty damn good. And playing the numbers, playing the odds, they tend to be right. However, the pathologist sees all the times when, the baby is reasonably normal. So the baby has been compensating. The placenta is not normal. The baby has no brain, above the brain stem, or has a massive stroke, or is dead, or is, badly damaged. So they play the numbers, and most of the time they figure if the baby is okay, the placenta has to be doing fine. And of every tissue that comes out of you in a surgical procedure, even a hernia sac has to go to the laboratory to make sure that the doctor did the procedure right. the placenta is the only tissue that comes out of somebody during a delivery or during an operation that can legitimately be thrown straight into the garbage. So, yes.
Because you really can't make these observations on humans in a controlled fashion, I mean, how is it that you are able to classify these various factors that contribute to low birth weight in a, objective nature?
By taking population studies and measuring those as carefully as possible. And, to be totally honest, there is a group of people who don't believe in what's called fetal programming, and they feel that it is impossible with statistical methods and regression and confounding and all of these other corrections that we can make, that all of what we see is just another way of saying that to be disadvantaged, socially disadvantaged, is a bad thing. So they don't believe any of this. They believe that it's all social disadvantage. Everything is outside of the womb. And all of this apparent association with birth weight is just that social disadvantaged children tend to be smaller. So there still is some argument as to whether or not there's any biology to this at all. And your question is an extremely good one, because and North Carolina has problems with their cohorts, because you can imagine that there's some bias in who agrees to give a hair sample and who agrees to have blood taken and what have you. And when I was in Chattanooga, we were trying to get fingernails and what have you, and to look for heavy metals. And we couldn't get anybody to do this because everybody watched CSI. And they were afraid we were going to run drug studies and send them off to the, nobody wanted to give a specimen, because they didn't want it, so yours is an extremely good point. So what you do is you try to do a population, not just one hospital or your own experience. You try to do things more broadly. And then you have multiple people in different parts of the world all trying to do the same thing, and you look at what might sort out of the background noise. And what I've given you is what tends to sort out of the background noise.
And then my follow-up to that is the human body is usually very resilient to abuse.
Speak for yourself.
What percentage of typically you would say bad mothers end up having healthy placentas and average or normal birth weight babies? That would sort of say skew your data or
Okay.
try to skew your data.
Well, one of the problems is it's hard to know. It's hard to know what birth weight you should have been. Okay? And people have asked me why I use cutoffs of less than 2,500 grams and greater than 4,000 grams. Well, those tend to be where the population distribution is about 10% here. Actually, now it's closer to 15, and about 10% here. Okay? So, and within that, every other birth weight is normal. So when you're asking me how many women who don't take care of themselves or who have other problems in pregnancy have babies whose birth weight is within the normal range, that's not the same question as how many women have babies whose birth weight is what you would have predicted them to be given the mother's birth. Do you see what I mean? Okay, so those are two different questions. And predicting ideal birth weight is problematic. There are a couple of algorithms that people have used that include the variables that I told you about, mainly maternal age, pre-pregnancy weight, paternal height and weight also kicks in. Half of the dad genes are in the baby, and dad genes are preferentially expressed in the placenta. Okay? Parity, and not only how much weight you gain from the time of your positive pregnancy test to delivery, but when in pregnancy you gain it. Okay? So all of those can be used to generate ideal weight. If you ask if I knew ideal weight, I would be estimating that probably between, I'd say 75% of women who have problems in pregnancy don't have babies with ideal weight, but I would say that are not ideal. But I would say that 85% to 90% of the birth weights are within the normal range. and thus don't give us any clues that that is a child that might be at some future risk. Okay? And that's what we're hoping that we can is improve outcomes. The best way, for example, if you are low risk, your chances of having a baby die after the 7th month before birth is 1 in 100. And if you're high risk, it's 1 in 1,000. Why? Because if we know to worry about you, we worry about you, and we can do something, and you have a lower chance of having the baby die. But if you're just walking around, what have you, you're 10 times more likely to have your baby die. So, you know, I failed IVF. My kids are adopted from Korea. They're twins, okay, really wonderful kids. But, you know, I was sitting here going, I'm going to be high risk. I'm also having my C-section at 35 weeks. But, you know, in any case, doctors were very relieved when I didn't get pregnant because they figured I was going to drive them absolutely apeshit. And they were right. Or they would have been right. But in any case, that would be my best answer to that. Okay? Once you're at risk, you can optimize your outcome because there's a lot of things that we can do. And right now, risk assessment is poor.